Hello and welcome to our channel. Today, we're diving into the world of immunophenotyping by flow cytometry, focusing on the CD8 test. If you're curious about how this test works and what it reveals about your immune system, stay tuned. Brief clinical history. Before we get into the specifics of the CD8 test, let's start with a brief clinical history. Immunophenotyping by flow cytometry is a crucial tool in diagnosing and monitoring various diseases, particularly those related to the immune system. This technique allows scientists to analyze the physical and chemical characteristics of cells, providing detailed information about their function and role in disease processes. Specification of the CD8 test. So, what exactly is the CD8 test? Let's break it down. The CD8 test is a specialized blood test used to measure the number and function of CD8 positive T cells in your immune system. These cells are crucial mediators of adaptive immunity. By identifying the presence and proportion of CD8 T cells, we can gain insights into the state of your immune health. Role of CD8 T cells. CD8 positive T cells, also known as cytotoxic suppressor T cells, play a key role in restraining certain types of immune responses. They are vital for defending against intracellular pathogens like viruses and for tumor surveillance, identifying and destroying cancerous cells. However, they can also contribute to excessive immune responses, leading to immune-mediated damage such as in autoimmune diseases. Diseases associated with CD8 T cells. Now, let's talk about the diseases associated with abnormalities in CD8 T cells. Abnormal levels of CD8 T cells can be associated with a wide range of conditions. These include test information, viral infections. CD8 T cells are crucial in fighting viral infections like HIV, where their levels can indicate disease progression and the effectiveness of treatment. Autoimmune diseases. Conditions such as multiple sclerosis and rheumatoid arthritis can show abnormal CD8 T cell activity, contributing to tissue damage. Cancers. Certain leukemias and lymphomas involve CD8 T cells, either in their proliferation or as part of the body's response to the cancer. By monitoring CD8 T cell levels, doctors can diagnose, monitor, and manage these conditions more effectively. Specimen used. The specimen used for the CD8 test is typically a blood sample. This sample is then analyzed in a laboratory using flow cytometry. But how does this process actually work? Flow cytometry analysis. Flow cytometry is a powerful technique that allows scientists to analyze the physical and chemical characteristics of cells or particles in a fluid as it passes through at least one laser. It's highly accurate and efficient, capable of analyzing thousands of cells per second. Components of the CD8 test. The CD8 test includes several components. Sample preparation. Blood is drawn and treated to isolate the white blood cells. Staining. Cells are stained with specific antibodies that bind to CD8 markers, making them visible to the flow cytometer. Flow cytometry analysis. The stained cells are passed through a flow cytometer, which uses lasers to detect and count the CD8 positive T cells based on their fluorescence. This comprehensive analysis helps in understanding the quantity and activity of CD8 T cells in the sample. Test report time. One of the great advantages of flow cytometry is its speed. Test report time is usually quite fast, often within 24 to 48 hours, which is critical for timely diagnosis and treatment planning. Importance of CD8 T cells in immune function. Let's delve a bit deeper into why CD8 T cells are so important. CD8 T cells are a subtype of T cells that directly kill infected or cancerous cells. They recognize small fragments of antigens presented by infected cells via MHC class I molecules. Once they recognize their target, they release perforins and granzymes, proteins that puncture the target cell's membrane and induce apoptosis, or cell death. Adaptive immune response. This ability to target and destroy cells makes CD8 T cells crucial for controlling viral infections and preventing cancer progression. They are part of the body's adaptive immune response, meaning they can remember previous infections and respond more effectively if the same pathogen is encountered again. Imbalance in CD8 T cells. However, an imbalance in CD8 T cells can lead to problems. Too few CD8 T cells can result in increased susceptibility to infections and cancers, while too many or overactive CD8 T cells can cause excessive immune responses, leading to autoimmune diseases and chronic inflammation. Monitoring and clinical significance. Monitoring CD8 T cell levels is crucial social and various clinical settings. HIV, IDS, CD8 T-cell counts are used alongside CD4 counts to monitor disease progression and response to antiretroviral therapy. Transplantation, post-transplant patients are monitored for CD8 T-cell activity to assess for signs of rejection or infection. Cancer and oncology, CD8 T-cell levels can be indicative of the immune system's response to tumors and the effectiveness of immunotherapies. By understanding CD8 T-cell dynamics, clinicians can tailor treatments to individual patient needs, improving outcomes. To sum it up, the CD8 test is a crucial tool for assessing your immune health. It helps identify and monitor various diseases, providing valuable insights into your body's immune defense mechanisms. This detailed understanding of your immune function can guide effective treatment strategies and improve health outcomes. Closing scene. Thanks for watching. If you found this video helpful, don't forget to like, share, and subscribe for more informative content. And if you have any questions, leave them in the comments below. Stay healthy and informed. See you next time on our channel, where we bring you closer to the science of health.